learned that you have actually worked on developing many different kind of molecules. The molecules having anti-cancer, anti-Alzheimer, antibacterial, anti-gout, and anti-lipidemic properties. So out of all these versatile molecules, which kind of molecule, or I would say which kind of project is very challenging for you, and why?
Okay, this is a really a tricky question because every research project, every drug discovery project is challenging. Not drug discovery means every science, like every research is challenging, right? So, um, but my particularly like I like the anti-Alzheimer project.
So
Why?
just to
Because
tell our audience, what is Alzheimer's? Like,
yes, okay. So Alzheimer is the disease condition. Do you know the dementia, right? The the partial or totally memory loss.
okay, so
That is
people
that.
don't remember things. That's what dementia is, right?
Yes, yes.
Okay,
So
okay.
people basically forget like normal things, right? So that is dementia. So Alzheimer is the bigger picture of that dementia. And the problem is why this Alzheimer disease is more challenging these days because we don't know exactly which proteins are really involved in this disease. There are a lot of targets has been found. Targets means proteins or enzymes has been found to be involved in this particular disease, Alzheimer disease, right? So no any drug therapy is the best treatment for the Alzheimer because some drugs are acting on some particular proteins or enzymes. Some drugs are on other targets. Some drugs targeting on the other targets, right? So that's why, like, we have the compounds which cannot fully treat this disease, but it can halt the progression of that disease so that we can prevent the further progress of that disease.
So in
So
this
why
case, if someone somehow, if we can find Alzheimer's in the early stage, then the compound which you developed are going to be very helpful because you can prevent them in early stage, and
in
then
early stage?
okay.
Yeah, yeah, that's the main thing. But the problem is the Alzheimer disease can happen like in the old age. So I believe the people over the age of 50 years, I think so, maybe 60 years. I'm not particularly remembering these things. But yeah, so the thing is, we have to develop, if we have to particularly treat this disease condition, we have to make the compounds which are multi-targeting. Means it can, uh,
Target
multi-targeting means
multiple enzymes,
multiple
you
targets.
mean? Okay.
Yes, yes. So by single molecule, uh, it should have the potential to inhibit or to target multiple enzymes or multiple proteins.
So
So
for
we
our, students, I'm what I'm learning is it is a very new field for now. If a student wanted to enter into the field to develop these multiple target drug molecules, so it is an emerging field, right?
yes, yes, yeah. So we tried to develop multi-targeting compounds during my PhD. Yeah. And fortunately, we got some of the hit molecules that can inhibit multiple proteins or enzymes, you can say. And those compounds behave better than the current treatment. Like donepezil is the current therapy for the Alzheimer disease. That can just halt the progression, but not fully treat. Yeah. So fortunately, those compounds, like multi-targeting compounds, are better than those current drug therapies, and also those compounds are non-toxic. Those compounds found non-toxic in animal model also. So I can say those molecules can be act as a hit lead molecules basically to further further optimization of the molecules, like especially for anti-Alzheimer, especially for Alzheimer disease. Yeah. So this was uh, my
One of the
favorite
most
project, I can
challenging
say. Yeah, most challenging, my
molecules you
favorite.
designed.
And
Okay.
this work was published in well reputed journal of American Chemical Society. Yeah.
Okay, okay. So
Yeah,
you know what? We will definitely gonna share few of your most highlight journals in the end, so that if some students wanted to read them, they can go there. Okay.
that would be perfect. Yeah.
Yeah. Thank you.
Okay, so you have used highly sophisticated tools like NMR, mass spectrometry, IR spectrometry, and UV spectroscopy,
Mhm.
Hector, COSI. So, can you help the audience to understand like how these techniques have helped you to in the drug discovery pipeline?
Yes. So basically, these techniques are to confirm the structure of the molecule. So suppose we are making some of the reactions, we are making some of the compounds by mixing two reactants to each other, and we will have the final product. And how would we know, like, this molecule is what we are trying to make? So we have to confirm the structure of that molecule. So these techniques like proton NMR, carbon 13 NMR, infrared spectroscopy, UV spectrometry, mass spectroscopy, these are basically the technique to confirm the structure what we have made. So proton NMR, carbon 13 NMR. So let's talk about proton NMR. The proton NMR tell us how many hydrogens are there in that structure and which kind of hydrogens are there. And carbon 13 NMR will tell us like how many carbons are present and what kind of carbons are there, like electron deficient, electronic rich, carbon 13 or not. And similarly, mass spectroscopy, as Ankur initially mentioned before this recording, the mass spectroscopy will tell us the exact molecular weight of our compound. It will show us the peak at the molecular weight. So, infrared and UV spectrometry will tell us the functional groups like hydroxy groups, like amine groups are there or not. So, all together, these techniques will confirm the structure we have made, or we are making, or we are trying to make. So, for training purpose, like to run these tools, to run these machines like NMR machine or mass spectroscopy machine, these are not challenging things to run the machines. So we have a computer software. You just have to familiarize with those interface of that particular tool or particular software, and you have to just inject your sample and just give the command, and it will run by itself. But to read those spectrum, like proton spectrum or carbon 13 spectrum, that is really challenging. That need I can say theoretical as well as practical knowledge. So this is like a mathematics. So more practice then means you will definitely get a more knowledge. Yeah.
Thank you
Okay.
for telling us this, Dr. Harvinder.
Yeah.
So, what I have learned so far, we first use molecular docking to understand what are our potential candidates, and then we have to make these molecules, and making these molecules is challenging. You have to do
Yep.
this structural characterization. But
Mhm.
once we have all these molecules, my next question is that I can see you have actually carried out both in vitro as well as in vivo biological
Mhm.
evaluations, including the animal studies. So, how
Yep.
was your experience doing these animal studies? And is this a very challenging to get expertise on, or is it a very exciting to working with animal models?
Okay, so yeah, in vivo experiment. So basically, I am a medicinal chemist, right? So this the to work with the animal model, that is some kind of pharmacology work, means other people, right? The pharmacologists usually do these kind of things like in vivo animal studies. But the thing is, like these days, like be you have to learn all of those techniques by yourself. So means you have to be combo of like a biologist as well as medicinal chemist as well computational chemist, right? So yeah, so I had to learn those techniques like in vivo animal studies during my PhD. Then I learned, and it was really challenging because uh, because suppose you are working in a medicinal chemistry field and you're running a chemical reaction, and after performing those things and you're directly switching towards the animal model, and you have to prick uh, the tail or eye. So basically we had to withdraw the blood from the eye of the mice or mouse by puncturing the retroorbital is the like is the vein in the corner of the eye of the mice. So we have to puncture that by the capillary to withdraw that blood. And it was really the challenging, like emotionally challenging thing for us to do that thing. But the thing is, uh, I can say every scientist uh, should emotionally strong if he or she is working in the research field, right?
Right.
So yeah, means we have to learn these things
Okay.
also. Yeah.
Actually, it's very, very, promising, like promising to the field that we have to be emotionally supportive, because nowadays it's not that much easy. For like being a girl, like I am actually scared of blood even. I can see the blood. So being a veterinarian or being, this taking up with the dogs and cats, so it's quite challenging nowadays.
Yeah, you should remember every time you should remember like why we are doing research, because we are like
Exactly.
for to save lives, to improve human health. So in that way, like we should have means we should remain emotionally strong in research
Yeah.
field. Yeah.